Hey, Patriot Biologist, this is lecture 2 for unit 1. In your notes packet, this will cover questions 8 through 14. So 8 through 14, where we left off from the last video, all the way through the science and engineering practices. So you will end with some example problems that we'll do in class in your notes packet. So here we go. Let's jump into it. Okay, so in science, again, the main goal in science is to try to come up with an explanation that is true to the best of our knowledge. So even when we have an experiment that doesn't go the way that we expected it to, or our hypothesis is not supported, that doesn't mean it's a failure. That means that you are learning how not to do something. Okay, so when it comes to these controlled experiments, that's what we're really looking at is not only are we looking to be right, quote unquote, we always like our hypotheses to be supported, but in these controlled experiments, we're also looking for ways that can make our explanation stronger. So with our controlled experiments, how we do this is we test a hypothesis using an experiment that keeps track of various factors that can change, or we call these variables. There's going to be a few different types of variables that we discuss during this lecture and that we do in class. So keep that in mind. A good experiment must be repeatable, not just by you, but also by others. So when you're designing an experiment, you're not only wanting to list every single step out to the best of your ability, but you also sometimes want these experiments to be practical. It's always best to start simple and work your way up. A controlled experiment, by definition, is going to be one in which we are testing a hypothesis using one variable that is changed. And in controlled experiments, we call this the independent variable. Everything else, every other aspect of the experiment must remain unchanged, and we call those constants. We can also call these controlled variables as well. So our controlled experiment, or our controlled experiment, we only have one variable changing. So in this particular example, this plant is getting water, this plant is not getting water, and what we would be looking at is the result of that, and that's what we are going to refer to as the dependent variable. Okay, so we're changing, this one gets water, this one does not get water. How does that affect the plant? So cause and effect is really all science is. All right, so some definitions in case you've missed them. The controlling variables, the variables that we're going to be working with here. The independent variable, we also call this a manipulated variable, so make sure we have both of those. This is the variable, this is the thing, the condition. This is what the scientist will change on purpose. This is the thing that they are purposefully changing so that they can see how the dependent variable or the responding variable, again, write both of those down. This is what we're looking at to see how is it affected by the independent variable. So if you're somebody who you really like English, and in English class you talk a lot about cause and effect, think of the independent variable as being like the cause, the dependent variable being the effect. Okay. So if we kick it back to our previous picture, in this example, our plants, if they get water or they don't get water, that is the independent variable. This is the thing that the scientist is changing on purpose. I am purposefully giving this plant water. I am purposefully giving this plant no water. As a result, we're seeing how the dependent variable is affected by that. So in this case, it looks like plant growth. This plant is growing. This plant is not. So our dependent variable is whether the plant grows, the green leaves, whatever it may be. All right, the control group is a particular set or it's a particular group, it's a particular part of the experiment that is exposed to the same conditions as the experimental group, except for one independent variable. Um, the control group is going to be the group in the experiment that remains constant. Okay, so for the control group, this is the group that does not get the independent variable. The experimental group does get the independent variable. Okay, so again, kicking it back to our plant, let's say that this is our control group, the plant that normally gets water. Our independent variable is changing that this one does not get water. And then with this one, we would see that when compared to the normal plant, this one is not growing as much. So that would be our dependent variable. But we do always, especially in a controlled experiment, we always want to have a comparison of, again, kind of a normal control group, what would normally happen, what would we expect to happen, and then compare that to what we're altering or what we're changing for the independent variable. Okay, so again, we compare the control is compared to the experimental. What normally happens will be compared to what the scientist altered or changed with their independent variable to determine how the dependent variable is affected. So in this case, sometimes our experimental groups, we can have multiple at the same time. I know this picture is kind of hard to see, but a control group, again, this is just getting regular old water. That's what we do. We're going to water our plant with just normal water. Our experimental groups are getting some sort of supplement. So one is getting nitrogen, one's getting potassium, one's getting phosphorus. And then maybe with that, again, we're looking to see how green the leaves are or how tall it grows and how quickly it grows, anything like that. So control group is normal. Experimental groups will get some sort of alteration, and then we will compare how they change to what our control group changed. And we can also compare each pot to one another. So we have a lot of comparisons, a lot of data coming in. Once we get done with our experiment, we do have to do some data interpretation or data analysis. When we do that, we in science say that the hypothesis was supported or refuted. We don't like to say right or wrong because, again, even if our hypothesis wasn't supported, there's still information that we're learning from that. So we don't like to say right versus wrong because that insinuates that there's negativity with it. It's supported, yes, our hypothesis went in the same direction that our experiment went, or the hypothesis is refuted. Our experiment went into a different direction than what we expected. There are two types of data, quantitative versus qualitative. Quantitative is numbers, so quantity, numbers. Qualitative is quality or a description, so things that we can't put a number on. So it could be things like color, smell, taste, texture. Whenever we're doing an experiment, we really want to try to have both types of data. The quantitative helps with putting together that data analysis, but the qualitative also helps scientists when you pass off your experiment to somebody else. It helps them to determine whether they're going in the right direction or not. And then some things we can't do an experiment on, so we use computer models. So we can't do an experiment on how an asteroid the size of a car, how that's going to impact the Earth because, well, we don't just have access to an asteroid the size of a car. And we wouldn't want to do that because the results could be cataclysmic. So instead, we can use a computer model to predict how that could happen. So again, here's our qualitative, just a diagram to help. You are welcome to put this into your notes to help you remember qualitative, colors, smell, taste, names, and your quantitative data, which is affiliated with numbers. All right, and then finally, the science and engineering practices. These are a list of methods that not only, so Millard Public Schools has adopted these, but this is something that we see across the country now. Science is going further and further away from just straight textbook information, and it's going more towards practical and applicable practices. So here are the science and engineering practices: asking questions and defining problems, developing and using models, planning and carrying out investigations, analyzing and interpreting data, using math and computational thinking, constructing explanations and designing solutions, engaging in argument from evidence, and obtaining, evaluating, and communicating information. So that is our science and engineering practices, and we'll talk a little bit more about these in class and how we apply them in early college biology. So that is it for lecture 2. If you have any questions, please don't hesitate to reach out. Otherwise, we will practice these and more in class, and I can't wait to see you then. Have a good day.